Welcome to Research Renaissance, presented by the Karen Toffler Charitable Trust. Embark on a journey into the forefront of cutting-edge brain science as we feature insightful discussions with a diverse array of guests, from early career researchers pioneering groundbreaking science to influential voices in investment communities, policymaking, and research institutions. Together, we delve into neurological diseases, their causes, treatments, and the quest for cures. Join us in advancing our understanding of the brain and addressing its ailments. Welcome to Research Renaissance. I'm Deborah Westphal, your host and guide through the fascinating future of the brain. Joining me today is Dr. Sudeshna Das, an Associate Professor of Neurology at Mass General Hospital and Harvard Medical School. We will be discussing Sudeshna's interest in the development of tools for multi-scale data integration from molecular to clinical data, and the application of data science approaches to the study of neurodegenerative diseases. Sudeshna is also a 2022 Toffler Scholar. So let's dive in. Welcome, Sudeshna.
Thank you, Deborah.
I'm very excited to have you on this podcast, and I'm really looking forward to hearing what you're doing and kind of getting caught up with some of the technology you've been working. Very exciting innovations, and I'm thrilled that you're here. I would like to just kind of start with looking back on your career. What motivated you to specialize in this biomedical informatics, and really kind of what keeps you passionate day to day?
Yeah, sure. So growing up, I had huge goals as like a 5-year-old. I wanted to cure cancer. So, but then when I went on to high school and math and physics and computation interested me more, and I joined engineering school and in India. And then after completing my undergraduate studies, I came to Boston to pursue a degree in biomedical engineering, focusing on applying computational tools to biology data. So I was one of the first few people who were trained, cross-trained in both like biology as well as and math and computation tools, and to be able to apply these tools to biological data. And that seemed like the perfect combination because it helped me, apply the tools and the methods that I enjoy, as well as being able to solve the problems in biology. And since then, I was hooked. I spent 10 years in a biopharmaceutical company using these techniques for like drug discoveries, specifically for cancer. And then when I came to Mass General Hospital, I have been focusing on the study of neurodegenerative diseases, specifically Alzheimer's disease.
So why cancer at the age of 5? That's kind of interesting. Was there somebody in your family that had cancer, or what was that influence?
Yes, I lost both of my grandparents to cancer, and had other relatives who were dying of cancer, and it was always a fear. What if my parents succumb to that? So at that time, when I was growing up, there were no medicines, and so that seemed like something that I wanted to do.
Yeah, so I'm sorry to hear that. What about being an engineer, a female engineer in India? Was it any different, or was it about the same for your male counterparts?
It was very different, for sure. So I was one of 10 students among 500 students.
Wow.
And so, yes. Yes, it was not very common for girls, women to choose to study engineering. But the group of girls that we all enjoyed, doing math and computing and engineering, and enjoyed solving problems. And, the 10 of us bonded together and are still friends. And, we even do work together sometimes. So yes, it was very interesting, and that's where I met my husband. And the 1 to 50 ratio, like 1 to 500 ratio worked really well. So there were lots of people to choose your two friends and life partners from. So that worked out very well. So I met my husband, and yes, we've been together since undergraduate.
That's great. That's a great story. So you said biomedical, that this was kind of a new field to integrate the biology and the engineering. How long ago was that? And is it still kind of a new emerging area of focus?
Yeah, sure. It's a great question, because I think I want to just like step back and do some definitions, right? So biomedical engineering is you're applying engineering principles for like biomedical use. And so it's a lot of it is about like building devices, like, EEG devices, or like imaging devices. It's also about engineering materials. But the field that like within that department, the field that I specialized in is called like computational biology. So you're basically a biologist, but you're using computation to like study biology. It's also called bioinformatics, and that is a term that people use more often these days to refer to like computational biologists. And so again, that was the field that started off in like around the 80s and to the early 90s, when physicists and mathematicians started looking at the DNA and the sequence of the DNA and the RNA. So DNA are the building blocks of life, and the RNA, like the genes that get transcribed into proteins, and those are the units within the cells in our body that do most of the work. So there's a lot of research that needed to be done to see like what is the human genome, and what are the collection of proteins, and the proteins, what are their structure and function. So physicists started like working with these with the structure of the proteins, and my advisor was one of the first people, along with my advisor Dr. Temple Smith, along with uh, Dr. Waterman, developed an algorithm to like compare the sequences of two proteins together with each other. And then if the sequences are similar, one can infer that their structures are similar. And if their structures are similar, that means you get an idea of what the function of that protein, right? So this work started around in the 80s, and I was one of his first graduate students, and very new at that time. So this field of computational biology and or bioinformatics started at that time. And but informatics is basically the field of curating knowledge, right? Curating information, right? Rather. And if that information is on biological data, then it's called like bioinformatics. But if it's with like clinical data, it's called like clinical informatics. And then people sometimes combine the two and will call it like biomedical informatics. So it's really the principles of like applying information technology and analysis to biomedical data.
Yeah, I appreciate that. That was a really good kind of description of all the different, because we hear all these different terms, and it seems like things are moving very quickly, especially with the technology. We hear about artificial intelligence or machine learning or big data, and I'm assuming all of those technologies are helping you with these, this curating knowledge or curating information on your specific topics, correct?
Yes, absolutely.
So if you need big data, and I would imagine you have a lot of diverse data sets. You mentioned the information on the genomics or clinical data that may be taken through doctors, or how is this integrating all this information to create that knowledge? Where are the challenges, and where are kind of the opportunities there for people like you to discover this new information?
Yeah, there are lots of data and lots of opportunities, as well as lots of challenges as well. So I'm going to start first with like big data versus like high dimensional data. So, large data sets. So large data sets, the technology that is producing in large amounts of data for biologists, they're called omics, right? So one kind of data is called like transcriptomics. So it's om is means like a collection in Latin, and so transcriptomics is the collective data on the gene expression of cells, right? So that's one kind of omics. If you're looking at the proteins and their levels, that's called proteomics. And there are others like epigenomics, and there are new kinds of omics that are being invented every day. So these are very large, high dimensional data sets. And as the technology is maturing, we are able to get this data at like finer and finer resolutions. So for example, transcriptomics, which is the gene expression data, we would get it at the level of the tissue. But these days, you can sort the cells that make up a tissue, and you can get the information about gene expression in each of these different cell types. So these data sets that we get are on the order of 2 million cells and 20,000 genes on each of these cells. So you can imagine what a large data set is. So these are what we call like large high dimensional data sets. And they are very structured, and you have a technology, and it's an assay that gives like a fixed kind of output. And on the other hand, you have big data, which is also high volume, but it tends to be like high velocity. So you're getting data at a much faster rate, and so it's a volume, velocity, and then it tends to be like unstructured. So you have to assess the veracity of that data. So a good example of that is like electronic health records. So I work with that kind of big data, which is real world data that hospitals are collecting, not for the purpose of analysis, but for providing patient care on their all the medical information that gets recorded, everything from their medications, their diseases and procedures, and hospital stays and so on, right? So that's a great example of big data that I use in my research, and it tends to be spontaneous and organic, and not really collected in a structured way for research. So other types of big data are like data on consumers for collecting information on like their preferences and developing algorithms on them. So that data isn't what we use in our research here. So these big data sets, as well as these large high dimensional data sets, both produce a lot of challenge, right? For the high dimensional ones, the biggest one is this technology keeps like evolving, and it's been like evolving like really quickly, right? We had single nucleus RNA-seq. That technology started about like 5 years back, and now we can measure at the single cell, not just like transcriptomics data, but like proteomics, and then the epigenome. So various kinds of data which tells us like how the gene and the genome is organized into chromosomes. And there's a lot of information and research that's up and coming that shows that this epigenome also plays a large part in Alzheimer's disease. And the epigenome gets, affected in aging. And it's really how the DNA is bound into the chromosomes and packed into structures called histones, and which parts of it is exposed and not, and how does that affect gene expression. So anyway, all these technologies are changing, and we get new technologies and new vendors that are producing these kinds of different technologies. And in terms of big data, again, the challenge there is the velocity and really the lack of structure in this data. So you have to go through and define control vocabularies and information around those. And so your question on, how do we approach and solving these problems? So really the primary role of an informatician, whether it's bioinformatics or clinical informatics or any kind of informatics, is being a translator, right? So we're trying to translate from one domain. So for example, on the clinical domain, where physicians will record a diagnosis code for billing purposes, right? So those codes, and there are lots of them, and different hospitals might use different codes, and the different practices might do. So if we want to combine those diagnoses with data that we get from research studies where information is entered in a very structured manner, diagnosis for Alzheimer's, we talk about, what's the stage? Are they cognitively unimpaired, mild cognitive impairment, or dementia, mild, moderate, severe, or and what is the presumed biology? Is it disease? And the physicians evaluate it with symptoms or with biomarkers. Is cognitive impairment a result of Alzheimer's disease or frontotemporal dementia, dementia with Lewy bodies, and so on. Right. So a lot of this work that we informaticians do is help to translate between research data collection or two different research studies with real world data sets, and helping to write tools that can transform the data from one to the other, so that we are able to get harmonized data sets and a way of linking all these different data sets together, so that we can perform computation on them. And then performing computation is the second step.
So I would imagine, so you've mentioned your focus now is on Alzheimer's and dementia. Do you have within the lab that you work and, in your leadership, do you have key questions around Alzheimer's that you're trying to answer, and then structure that data and that, data analysis around those key questions?
Yeah, absolutely. Yes. We start with a research question. And right now, I'm working with Dr. Hyman, who leads our Alzheimer's Center at Mass General Brigham, and Dr. Alberto Serrano Pozo, who is also a Toffler Scholar, in understanding the roles of the different cell types in Alzheimer's disease. We are investigating that question using this single cell omics data. And we want to know, are the changes linear? If you look at donor brains across the Alzheimer's disease continuum, from healthy controls to the most advanced disease, is the progression linear, or or are the patterns of progression different? So in this large study of like 80 progression, we have been studying, for example, Dr. Serrano Pozo and myself have been leading a study on a type of brain cells that are called astrocytes. And because of their pretty, star-like shape, and these cell types in the brain, their primary function is to maintain homeostasis in the brain. So it's really these cell types need to function and do their work. But in Alzheimer's disease, what we have found is that they're affected, and their role in synaptic signaling. So this is an important component of the neurons firing and passing messages, and really for the brain to work. And we find that this synaptic signaling is affected in Alzheimer's disease. And then, interestingly, what we also found is that these astrocytes first, fight and mount a response to fight against the Alzheimer's disease pathology. But then in end stage disease, they're burnt out, and we observe this phenomenon of exhausted astrocytes when they have been exposed to pathology for a really long time. So we were able to recently publish these findings in a Nature Neuroscience paper, so which is forthcoming. So we were very excited about that, and we are also studying. This is how microglia and endothelial cells all work together with the neurons in the normal healthy controls. They work together as a functional happy family in and there's brain homeostasis maintained, and lots of things go wrong in the Alzheimer's disease. So our next step is to go from looking at individual cells and the genes within them to see how these cells work together in brain tissue. And that's where we need more information and more advanced multi-scale models and more technologies beyond these single nucleus RNA-seq or transcriptomics data sets to something that's called spatial transcriptomics. It's another new type of technology and tool that we are using to investigate these questions of how do all these cells work together in the normal aging brain and what goes wrong in Alzheimer's disease.
Yeah, that's interesting. And this might be a really silly question, but if the astrocytes get, and if they're fighting the disease and then they get fatigued, they get tired, and then they die, is that, is that the potential for some sort of therapeutic that would be used to keep those astrocytes fighting? Or, is that, do I understand kind of, because you're doing the kind of the understanding of the why is this happening inside the cell? Somewhere down along the way, there's going to be some sort of therapeutic, whether that be a drug or vaccination or whatever it may be. Does that make sense? Is that too silly?
No, it's definitely, that's the ultimate goal. Is we do want to understand what are the changes in disease, and so we study disease mechanisms. But the ultimate goal is, by understanding the disease mechanism and the evolution, can we develop therapeutics that can reverse disease, right? And Alzheimer's disease, there are two main pathologies, which we is called the amyloid and tau pathology. In amyloid, you find these little clumps of amyloid, which are called plaques, outside in the brain, but outside the cells. So they're extracellular plaques. And as well as tangles. And these are deposits of the tau protein inside the neuron. So that you can actually see in neuropathology under a microscope these pathologies in the Alzheimer's brain, right? So yes, the astrocytes we see have stopped performing their normal brain function, and the brain homeostasis is disturbed, and that gives rise to symptoms. So how can we reverse that? It's a really good question. And the main, the focus of curing Alzheimer's disease is to remove this the plaques, right? To actually change the disease is to remove the plaques. And so, like now we have like 3 drugs on the market that are approved by the FDA to remove plaques. The idea is that if you remove the source of the problem, then everything will go back to normal. It turns out it doesn't quite, because once the damage is done, it's like really hard to fix it. So, for example, like in cancer, you're trying to stop the cells from dividing, so you're trying to kill cells. But here in Alzheimer's, the cells are dying. So how can we revive them? And that's a much harder process than like rejuvenating and giving life is much harder than like killing, right? So the approaches that people are using as one of the things that you mentioned are like vaccines, right? Can we put in like challenges, right? Can we challenge, like just like in a vaccine, like for the COVID vaccine, you challenge your immune system with small amounts of a pathogen, and then your immune system learns to fight that infection better, right? So can we train our immune system to fight off these plaques, like and that's much safer than like a drug going into the brain and trying to clear the clumps, because when you're doing that, you might cause like damages to the blood vessels, or you can think of like that's a much more invasive process versus like your own body. If it can fight off those plaques, then it's a much a better way to get rid of the plaques or the tangles than artificially trying to design a drug that holds on to the drugs and clears it. So yes, so vaccines are coming, and I was involved in a study with the BCG vaccine, which is used for like treating tuberculosis worldwide. In the US, it's approved by the FDA to treat bladder cancer, and turns out that the BCG vaccine, which is not against, designed against amyloid, but rather the bacillus. It's really hard for me to say the word. Like it's the TB bacteria that it's designed for. It does give the immune system a boost. So we have profiled cells, and we see that after patients are treated with BCG vaccination at baseline and 3 months, you see a stronger immune system that is able to fight the brain invaders, so to speak. Yeah, so this is very much work in progress, and one other technology that you mentioned before is the use of AI. When we build these models, the way that AI is progressing, we can build these models, and then we can play like in silico experiments. Like, okay, what happens if we knock out this gene? How does that affect astrocytes and its interaction with the neurons and the microglia? And eventually to see, does it have an impact on the disease pathology?
So you mentioned a term or a new technology called spatial transcriptomics. I believe you pronounced it that way. What is that?
Yeah, so spatial transcriptomics studies gene expression. So transcriptome is the gene expression, and we are looking at gene expression. And it's very similar to the other one that I mentioned, which is single nuclear transcriptomics, where you can look at the gene expression in each cells. Here with spatial transcriptomics, you can look at the physical map of these gene expression, right? So if there is a plaque, we could look at the microglia, which is another type of cells around these plaques, or within the neurons and the astrocytes, which work together to conduct these signals. With spatial transcriptomics, you could look at the gene expression of the neuron and the astrocytes and get a map of the gene expression data for the whole brain tissue. And this is really exciting because earlier we could measure this gene expression only at the tissue level, right? We could look at the microscope and look at the plaques, but really not be able to look at like inside the cell and the genes and their levels within the cell. So with this technology, we are able to get a much better, like a physical map of where these gene expression are relative to the Alzheimer's pathology. So it's kind of like if you have the map of the United States, you can get like a state level data, but with these new technologies, we are getting town and county level data to data inside the household, right? So like we can look like inside the cell and get like subcellular resolution. So granularity and the resolution keeps like increasing day by day, and this particular technology is really exciting and promising because you can look at the changes where the action is, right? Like where the neuropathology is. So it allows us to hone into these special areas and give us a look into the brain at a resolution that we have never been able to do before.
Yeah, that's fascinating. And is that all visual? Is it that visual look? Is it pretty stationary, or can you see, because if you're using brain tissue that's in like the brain banks, I would imagine that's kind of stationary. But is there a possibility to see movement down at that real detailed level also about what is going on inside the cell?
Yeah, that's a great question and a fascinating one. So colleagues of mine, Ted Zwang and Rachel Bennett, and Ted's also a Toffler Scholar, have been doing this really fascinating work in animal models. So these are mouse models of Alzheimer's disease, and as well as like control mice. So they make a little opening in the skull and do it in a way that is, causing minimum pain to the mice. And they have, with the microscopes, they're able to look at how these neurons or any other cell that they tag from day to day in these animal models. And it's really exciting to see. And Alzheimer's disease is a disease that changes and progresses in along like months and years and not like days and weeks like in people. Even in animal models, you don't see changes in like day to day. It's like more like months to months before there's enough accumulation of pathology in these animal models. So it needs a lot of more more patience than like if you were studying cancer, where like the cells are duplicating in the order of days rather than like months. But yes, we could look at these changes over months in these animal models, and the way they make sure that it's the same cell that we saw like a month back is the architecture of the brain vessels that are like going through the brain. So you can get like a little like a zip code of like, okay, here's this one where these two vessels are branching, for example. So then you can see that, oh, okay. And this is the same neuron that I had studied a week earlier. And you can follow this over days and weeks, up to I think a month in these animal models.
Yeah, that's super cool. So you talked about the breakthrough that you and Alberto have kind of published on, I guess, and that publication is going to come out. And it's this role of astrocytes in keeping everything kind of healthy and fighting the disease. What other kind of breakthroughs or kind of insights are have you had, or just things that you've been really excited about more recently or just in your past that around this understanding of Alzheimer's and dementia?
Yes. So one is more current and the other one is in the future. I mentioned our work with a trial of the BCG vaccine. So as part of the trial, the investigators who are conducting the trial are profiling cells from our cerebrospinal fluid, so you can get a spinal tap. And these fluids bathe the brain, right? So the data and the cells that are in the CSF are kind of getting a little brain biopsy, if you will. So it's a little teaser of okay, here is what might be going on in the brain. So, and CSF assays are done frequently for measuring Alzheimer's disease. So in these CSF cells, what we found are a certain kind of immune cells. They're called T cells. And the goal of the T cells, they're in your white blood cells in the blood. And their goal is in adaptive immunity. So they, when they see a particular antigen, their job is to tell the killer cells, look here, this is something going wrong, and you should kill these particular cells because they're infected, right? So what we have found is that these immune cells, and particularly these T cells, have a large role in Alzheimer's disease, right? And that's fascinating because these cells are in the blood. With these latest technologies, we found that they're present within the brain tissue as well, and really play a large role in Alzheimer's disease, which we can get a peek into from profiling these CSF cells, but also by profiling the brain cells, because we do see a few of these T cells that have leaked into the brain tissue and creating havoc there. And although their primary function is surveillance and get rid of antigens, but they may be getting hyperactive and overstimulated and start attacking neurons and giving them killer signals. So understanding the roles of these, and then again manipulating these cells as a way to treat the disease, are both really very exciting approaches.
Yeah, there seems to be more kind of emphasis on that cardiovascular versus, in conjunction with the brain through that blood-brain barrier. So am I understanding that right? Is that there's these T cells and they're sneaking into the brain through the blood-brain barrier somehow, and we don't know why they're there and what they're doing. That's kind of what you're saying. Is that correct?
Yeah, no, and there's recent work that have been done at MGH and at other places, and as well as work that I've done in my lab, which shows that they are indeed getting more frequent in the brain tissue as the disease advances. And so more work needs to be done, how exactly are they toxic to the brain, and once we have an understanding, then start designing how can we manipulate these cells to do the opposite of killing the cells.
Yeah. So you'd mentioned a future breakthrough that you're pretty excited about. Can you tell me about that, and want to share anything about that excitement?
Yes. The technology that I'm really excited about is artificial intelligence tools and how to apply them to understand and model the disease, as well as do in silico experiments. The data that I have been describing is really complex. So, we are getting all these omics data from different kinds of technologies, uh, as well as other kinds of data from big data. And one particular kind of AI model that I'm particularly invested in are called graph neural nets. And what we are starting to do is build these relationships that we observe from omics data or from other kinds of data, and model them as a graph, which are basically circles or nodes, or which represent entities like genes or cells, and edges, which is a line between these circles that can represent the relationship between these different entities. So we can create this graph from all these, heterogeneous, multi-scale data. And once we have created this graph, we can do this training where this graph is able to learn from the underlying data structures and patterns, and come up with representations, which are embeddings or numerical vectors to represent each of these relationships and entities. And by developing these large models, then we can use them for a wide variety of downstream tasks, everything from in silico predictions of knockouts, okay, what happens if we tweak this gene and we delete it? Or what happens if we control this gene expression and lower it by half? Or what happens if we stop the astrocytes from communicating with the neurons, or rather the T cells that can be cytotoxic, from giving the killer signals to the other cells in the brain? Or how can we curb these cells that are going wrong and stop the process, but also encourage the process that is able to clear the plaques and tangles from the brain? So using these models, we can really start asking these various questions about what happens if we manipulate them. And in the future, as we do that, we can also, on these graphs, take an individual patient's data, right, that we get from them from using either omics or lots of different data sets, throw this on the graph, and then tailor the graph to predict what is the key pathways to disease for this particular individual, and what drugs may work to cure their medicine. So I'm really optimistic that being able to model this vast amounts, and we're really talking about millions, if not billions of data points. How can we organize all this data and compute on them? And AI seems to be the technology that's ripe right now to leverage to solve this particular problem, where one day, hopefully, we can get the right drug for the right patient at the right time.
Yeah, that would be incredible. That kind of personalized care, personalized therapeutic for the individual. As you were talking, I was thinking about the time that if you were to do all of that without AI, it's almost virtually humanly impossible to manipulate those kind of what ifs. What, as you kind of just a little bit into the future, maybe 3, 4, 5 years, just kind of hypothesize how fast would you be able to take someone's data, both, internal data, cellular data, and then maybe even the the epidemiology data, or the, not the epidemiology, but the outside data, their pattern of life data. How fast could that, are we talking a year? Are we talking months to kind of come up with a vector, if you would, for treatment for a person?
So I would say that, within 5 years, we are able to combine the data, like you said, not just their biology data, which we can take like maybe a drop of blood, we sequence, or we take some sample from their CSF to get an idea of what's happening in the brain. We can get other real world data of their life experiences. We also get, and I work with data from like wearables, so like an Apple Watch or a Fitbit, for example, so you can get an idea of what's going on in their life and their environment, or sensors at home, and to be able to combine and do predictions of decline, and be able to also predict like the pathways that are going wrong. In terms of the therapeutics, I think that's going to take longer because drug development has a process, that has to go through first a development and an optimization to like a clinical trial before it gets approved. So that process takes always a little bit longer. So I would say that within 5 years, we should be able to have these models that can take different kinds of data. We call them multimodal or multi-scale if you're calling, cells to tissue and brain level data, combine them to make predictions. Can we, in 5 years, get the drugs as well? Maybe. I would love to have that happen because, the AI models are also being used for drug design and rational drug design. And that process is being sped up by AI in a way that is that's incredible to think about. And a student of mine who's an undergraduate wants to be able to tackle all this problem by the time he graduates next year.
That's great. That's great. How do you mentor? I mean, these really bright, energetic, undergrads, and because I know mentoring is really important to you, to these young researchers. How do you do that, and kind of what advice do you give students who are just kind of starting in this field of medical informatics?
So I have a pretty much an open door policy. So people approach me from high school all the way to PhD students and postdoctoral fellows. And this person who I was talking about first approached me when he was 14. So I've been like working with him since for a third of his life, and he's like around 21 now. So what I do is I ask a few questions. I want to see if like you are motivated and you are really excited about working on this problem, and to show a commitment that I want to spend part of my life doing this, because of course like high school and college students have their main goal, which is to be a student. But if they show motivation and they're willing to learn and they have the ability to learn, then it doesn't matter if they know it or not. So I'm happy to teach them. I currently have like a dozen students, and I work with them over the weekend or in the evenings. Like we have meetings, and post COVID, Zoom has really helped us, connect and be able to work with them. And because all the work that I do is on the computer, there are no barriers. I have like a couple of students from like Texas and California, so like physical location isn't a barrier for us. So I really love mentoring, and I have a lot of students, and they either do like a high school project and are able to use that and get an idea of what they want to do for their undergraduate, to like postdoctoral fellowships, and to all my trainees. I have some simple rules. First is figure out, what you want to do and who you want to be, right? Like just figure that out. Like, what is it that motivates you and excites you and that you'd love to do, even if you're not getting paid. I'm not advocating that you don't look for for a salary and maintain, but you want to do this because you love it, right? So figure out your passion and then make a plan. Like, what do you want to be like, 20 years from now? These are for the high school students. And then work backwards. So what does that mean 10 years from now to 5 years to year to work backwards to this week and this month, and then look at things and pick and choose and see, like, is what you're doing getting you towards your goal. So that's one of the principles. The second one is more about productivity and managing different activities to focus on things that are important rather than urgent. And this is from like Stephen Covey's book that I learned from back, when I was a manager, like a junior manager at a pharma company. And the idea is that don't let things that seem important, and urgent, like responding to an email right away, distract you from the things that are important. And what he says, and this is really true, if you focus on the things that are important, then over time you'll have less and less urgent issues, because you would have thought through and planned your life better. And the third principle is, above all, have fun, because nothing is worth it if you're not having fun.
Very good advice. I can't imagine being 14 and, asking for help and mentorship and wanting to be exposed. That's incredible. And good for you for making such a big impact in these young people's life, because, so many people don't really have those role models, and then they kind of wander around a little bit. But, boy, giving focus to a 14-year-old, that's pretty amazing. So, you're doing a lot of really complicated things and a lot of things that are really kind of cutting edge that we haven't, that researchers haven't done. It's just not out there in the market. It's not commercial. Is it hard to get funding and attention sometimes from funders to help you along in your research? And what do you do to try to keep that support?
Yeah, no, that's a great question. It's kind of like building your own startup, right? And you have to bootstrap. And to be able to do that, you have to do your work as well as like look for funding. And to help with that process, seek the help of mentors, right? So I have a wonderful mentor, Dr. Hyman, and he has helped me show the ropes, and and how to get funding from the NIH, which happens to be like one of the largest source of like biomedical research funding. But to able to get an NIH, investigator award, like which is called an R01, you need like preliminary data, and that's where we are incredibly grateful for foundations like the Toffler and other foundations that are willing to invest in some key experiments and new technologies and unproven ideas in the Wild West that can help us get some preliminary data with which we can apply for NIH funding.
So as we wrap up here, you talked a little bit about the future, and you could tell that you were excited about some breakthroughs. What is your greatest hope for the future? When you look out that 10, 15, 20 years, what is your greatest hope?
Yes, I would love for a day when we have treatments that reverse Alzheimer's disease and do it in a safe and effective way, and that we are able to find this the right drug and the right time for a particular individual. So, precision or personalized treatments. And if AI and omics and all these tools can help accelerate this process, nothing can give me like more satisfaction than having made a tiny contribution to that process.
Yeah, it sounds like you're right in the middle of that contribution too. As you said, you're kind of translating, and you're that translator, and the technology is moving so fast that that future hopefully will come at us a lot quicker than what both you and I can imagine. Sudeshna, this has been amazing, and I could listen and pelt you with questions for many hours here. But if people are interested in what you're doing and finding out more about what you're doing, or want to collaborate with some of your research, how would they get in contact with you?
Yes, you can reach me at my email is das, S as in Sam, D as in David, A as in Apple, S as in Sam at mgh.harvard.edu. I answer to most emails within a week or so. I try to get back to people, so that would be the best way to get in touch with me. If you are also search my name in the Mass General Brigham website, you might find my contact information there as well. And so the website, it's a little bit older, so not all information there is current, but email and the contact information on the website should be up to date. So that's a great way of getting in touch with me. So anybody who is interested in using the latest tools and technologies for combating Alzheimer's disease, I'd be very excited to hear from you and help you figure out a way to see if you want to and how to work on that. And I'd like to start by thanking you, Deborah. This was wonderful, and it was such a pleasure talking with you. But I do want to say that I feel that my contribution is like a small cog in the wheel. There are so many smart people and so many dedicated researchers working to this. So if I can help make a tiny contribution, that makes me happy.
Yeah, well, you're very humble, and I do really appreciate what you're doing. And yes, there's a lot of people out there working on this, but it's they're horrible diseases. And just more and more people are in contact with these diseases. And so thank you so much for, the time today, and then just your dedication for what you do on a day-to-day. So it's been a real pleasure, and thank you again.
Yeah, thank you. It's my pleasure.
Thank you for joining Research Renaissance. For more information and to stay updated, subscribe for more episodes, and visit our website at tofflertrust.org. Feel free to call us if you have any thoughts, suggestions, or just want to find out more about the Karen Toffler Charitable Trust. Until we meet again, onward and upward.